Let us discuss the color joint exam biology paper 1. On to our first question. Name 2 enzymes in the human digestive system which are secreted in an inactive form. Okay, so we have pepsin, its inactive form is pepsinogen. Trypsin, its inactive form is trypsinogen. And lastly, renin, whose inactive form is prorenin. Part B. State the term used to describe such enzymes named above. So enzymes that are produced in their inactive form and activated when needed are referred to as precursors or zymogen. Explain why the above named enzymes should be secreted in an inactive form. Okay, let me explain. Now, enzymes are crucial for a lot of biological processes such as digestion of food substances, but they need to act at the right time and place to ensure, of course, proper functioning of the body. So, by secreting enzymes in an inactive form, the body can then regulate when and where they become active. So, for example, a digestive enzyme such as trypsin, this is produced in the pancreas, but it functions in the duodenum. So, if trypsin was to be secreted in its active form, what do you think will happen? It's going to start digesting proteins because trypsin acts on proteins. And where will it find proteins? In the cells of the pancreas. So it will start breaking down, digesting the tissues that make up the pancreas, which of course is very dangerous because it will lead to tissue damage. And this is something that cannot be allowed to happen. So they are secreted in their inactive form and only activated when needed. Question number 2. List 2 importance of studying biology in high school in today's world. Number 1, it helps in acquiring scientific skills such as classifying organisms, recording, measuring, analyzing, etc. Entry into careers such as medicine and agriculture. It also helps us in solving environmental problems such as food shortage, poor health services, pollution, and others. Question number 3. Part A. Name 2 theories that explain the origin of life. We have the special creation theory and the chemical evolution theory. List 2 methods used by scientists to determine the age of fossils. So we have carbon-14 dating and potassium-argon dating. Question number 4. The diagram below shows members of a certain group of organisms found in a certain habitat. Use them to answer the questions that follow. Okay, so we have organism W, this is a dolphin, so a mammal. Organism X, this looks to be a frog, so an amphibian. Part A, name the phylum into which the above organisms belongs. These are vertebrates and therefore they belong to phylum Chordata. Part B, name the class into which organism W belongs to. This is class Mammalia. Part C, explain why the circulatory system of organism X is said to be inefficient in oxygen transportation. Okay, now the circulatory system of amphibians such as the frog is considered to be inefficient in oxygen transportation largely because of the way their hearts are structured. Now, when it comes to mammals and birds, we have a 4-chambered heart. So, 2 atria at the top and 2 ventricles at the bottom, where you're going to have oxygenated blood and deoxygenated blood completely separated. No mixing of these 2 types of bloods. Now, when it comes to amphibians, such as the frog, they have a 3-chambered heart, which consists of 2 atria and 1 ventricle. So the right atrium receives deoxygenated blood from the body, while the left atrium receives oxygenated blood from the lungs and the skin. And yes, the skin. Remember, some amphibians like the frogs use their skin as a respiratory surface. So all good, right? So in the atria, you have oxygenated blood in the left, deoxygenated blood in the right, separated. But the issue comes into with the ventricle. Remember, they only have 1 ventricle. So you're going to have oxygenated and deoxygenated blood mixing here. Now this mixing reduces the overall oxygen concentration of the blood that is being pumped to the body. So as such, the tissues are going to receive blood that is not fully oxygenated, which makes the whole process less efficient. Now, I want to say this. In some species of amphibians, such as the frog, they have a valve that is present within their ventricle. Now, as stated, they only have one ventricle, so you're going to have mixing of blood. But in order to minimize this, they have a valve. Now, the valve, provides some degree of separation of the two types of blood, but mixing still happens. So how do we phrase our answer? We're going to state that they have a partially divided ventricle, therefore oxygenated and deoxygenated blood mix. So it makes the whole process less efficient. Question number 5. In radishes, red and white are pure breeding colors, while hybrids are purple. If a red radish is crossed with a white radish, what will be the phenotype of the F2 generation, assuming the F1 generation self-pollinates? Use letter R for red and W for white. Now, before we start, that is a radish, just to have the picture in our minds. Now, when it comes to the information we have, we've been told that we have pure breeding plants and hybrids. So let me define what these two are. Now, when it comes to pure breeding plants, these are homozygous for a specific trait. That is, they have two identical alleles. Now, in this case, in our radishes, we have three colors: red, white, and purple. So, our red and white plants are the original parents, okay? And these are pure breeds. So that means what will be our genotype for the red radish? It's going to be RR. Why do we have two R's? Because this is a pure breed. Amanisha, it's homozygous, so two R's. When it comes to the white one, this is also a pure breed, okay? So we are going to have two identical alleles, WW. Now the offsprings, okay? These are the F1 generation, are going to be hybrids. They are going to have one allele from the red radish and another allele from the white radish. Now, hybrids in this case have a distinct color of their own, which is purple. So, their phenotype is purple. What is their genotype? RW. Now, the question is not asking us for the genotype or phenotype of the F1 generation, but for that of the F2 generation. So, what will happen is that we are going to take the F1 generation, cross them, because we've been told that they are going to self-pollinate. So, we are going to take two members of the F1 generation with that genotype and that phenotype, cross them in order to get our F2 generation. So, when we cross them, those are the genotypes that we are going to get. So, as you can tell, we have three genotypes. That is responsible for the red radishes. The second one, those are the hybrids. So, we're going to have a purple radish in this color, two purple radishes. And the last one, of course, is going to be white. And those are the phenotypes of the F2 generation. Question number 6. The diagram below shows a cell as seen under an electron microscope by a student at Kala Secondary School. use the diagram to answer the questions below. Okay, so on the diagram, label the structure that represents mitochondria. So mitochondria are sausage-shaped. They have four rings called cristae. If we look at it, we can easily pick out where the mitochondria are. Part B, name two structures visible with a light microscope which distinguishes plant cells from animal cells. Now, look at the question. The question is not asking you whether this is a plant cell or an animal cell. No, it wants to know whether if you have a cell and you're observing it under a light microscope, which two structures are you going to use to determine whether this is a plant cell or an animal cell? Number one, the presence of the cell wall. So if you have a cell wall, that makes it a plant cell. If the cell wall is absent, you have an animal cell. The next is the nucleus, specifically the placement of the nucleus. Is the nucleus centrally located? Then yes, that is an animal cell. Or is the nucleus located at the periphery, near the edge of the cell? That is the plant cell. Now when it comes to plant cells, most of the space is taken up by the vacuole. The vacuole is large and it's centrally located. It's at the center of the cell. So the nucleus is pushed towards the edge of the cell. So the nucleus. Now some of you might say chloroplast. And in most cases, yes, but I want you to remember that not all plant cells have chloroplast. So you might end up having a cell that doesn't have chloroplast, but it is still a plant cell. Let me give an example. Root cells, okay? So the cells that are present in the roots of plants don't have chloroplast, because why would they have chloroplast in the first place? These are cells that are underground, so they don't even have access to light. They don't have light at all, and chloroplasts only function in the presence of light. So yeah, there's no need for them to have chloroplasts. Another are the epidermal cells that are found on the upper and lower surfaces of the leaves. Apart from the guard cells, none of them have chloroplasts. So please don't use this as your answer. Moving on to the next question. In an experiment, a student observed the following under a light microscope after placing a transparent ruler on the stage, followed by placing a slide containing onion cells. Estimate the cell size in micrometers show you working. Now for us to estimate the cell size, we are going to use this formula. Diameter of the field of view in micrometers over the number of cells along the diameter. Now, when it comes to the diameter, we are going to use the ruler markings to guide us. So what is the diameter of the field of view? So we have 1, 2, 3, 4, 5, 6. So that is 6 millimeters. Sawa? Now we need to convert this to micrometers. 1 millimeter is equivalent to 1000 micrometers. 6 will give us 6000 micrometers. How many cells do we have along the diameter? So if we count the number of cells, it gives us 6. So that means the size of one cell is 1000 micrometers. Question number 8. Distinguish between epigeal germination and hypogeal germination. So in epigeal germination, cotyledons are brought above the ground level, while in hypogeal germination, the cotyledons remain below the surface. Part B. Name two regions where secondary growth occurs in plants. Now when you talk about secondary growth, this is responsible for increasing the girth, the thickness of the plant stems and roots. Now, this process mostly occurs in dicotyledonous plants and gymnosperms. It's very rare in monocots, okay? So when you talk about any type of growth in plants, this occurs in the meristematic tissues. Now, meristematic tissues are of two types. We have the apical meristem and the lateral meristems. Now, it's actually the lateral meristems that are involved in secondary growth. Apical meristems are involved in primary growth. So primary growth brings about the increase in the length of the shoot and the roots, while secondary growth increases the girth of the plant, upana wa ule plant. So as stated before, this is as a result of the lateral meristems. Now we have two types of lateral meristems that are responsible. So we have the vascular cambium and the cork cambium. And those are our answers. Question number 9. The diagram below shows part of the alimentary canal in animals. Study it to answer the questions that follow. Part A. Identify the structure drawn above. That is a villus. Part B, Roman 1. Name one common protozoan disease that affects humans in the part shown above. This is amoebic dysentery. Roman 2. Name the causative agent of the disease named above. So, the cause of amoebic dysentery is Entamoeba histolytica. Question number 10. Study the diagram below and answer the questions that follow. Okay, so we have a diagram showing us a baby suckling from the mother and a section of the breast tissue. So part A, name the hormones responsible for the following processes: milk secretion, prolactin, milk letdown, oxytocin. Okay, now let me explain. So what happens is that in this case, we have two things, okay? We have the secretion of milk. This is the production of milk by the mammary glands. And then we also have the release of milk. So once the milk has been produced, it is then released, into the baby. So yeah. Now, I'm going to explain how each of these happens. So what happens is that when labor is near, when childbirth is near, the level of progesterone in our body drops. Okay, sorry, in the mother's body drops. Now, progesterone is maintained consistently at high levels during, but towards the end, when labor is near, the level of progesterone drops. So this sends a signal to the pituitary gland to release prolactin. Now, prolactin is a hormone that stimulates the mammary glands to start producing milk. Sawa? So, milk secretion, milk production is as a result of prolactin. Now, let's talk about the milk letdown reflex. Now, when you talk about the milk letdown reflex, it's the release of the milk. Now, this is done based on certain stimuli, like suckling of the nipple by the baby crying, or even the sight and smell of the baby. All of these can bring about the release of milk from the mother's nipples. So when these stimuli happen, you're going to have the hypothalamus sending impulses to the pituitary gland to release oxytocin. Okay? Oxytocin then acts on the mammary glands. So it stimulates the contraction of the muscles in the breast tissue. These contractions force milk. Remember, the milk has already been secreted, it's already been produced, it's there. So we want it to come out. So these contractions force the milk into the milk ducts, where it then travels down to the nipple for the baby to feed. So while prolactin is responsible for milk production, oxytocin triggers the release of the milk. Roman number 3, state one condition that may inhibit milk letdown. So this reflex can be interfered with by emotions, okay? So I'm talking about things like anxiety, distraction, or even if the mother is feeling embarrassment, you can have situations where the milk is not being released, okay? So it interferes with the impulses being sent to the pituitary gland and oxytocin is not released. So milk does not come out. Question number 11. A student drew the diagram below after carefully observing a certain process in a school botanical garden. Use the diagram to answer the questions that follow. Okay. So this is double fertilization in angiosperms, in flowering plants. Now I want to say this. I'm not going to go into detail regarding what happens during double fertilization. There's a previous video of mine. It's one of the prediction papers for either biology paper 1 or biology paper 2. I'm sorry, but I've forgotten which it is, where I discuss in details You know how double fertilization takes place, how you're going to have the endosperm, how you're going to have the zygote being formed. So if this is a topic that is challenging for you, please check it out. Part A. Identify the type of cell division that takes place around region labeled B. This is mitosis. Part B. State one role of the structure labeled D. So D is the pollen tube. So it facilitates the transfer of the male gametes from the stigma to the embryo sac. Part C. What is the fate of cyanogens and antipodal cells after fertilization? So these two simply disintegrate. You know, they just break down because they have no further purpose after fertilization. Now, when it comes to cyanogens, these are two. So their purpose, their role is to guide the pollen tube towards the egg cell so that fertilization can take place. So how do they do so? They release certain chemical signals that attract the pollen tube to where they are. So when the pollen tube reaches the embryo sac, the cyanogens simply break down. So they do so in order to provide an opening for the male gametes to enter and fertilization to take place. Now when it comes to the antipodal cells, these are three and they provide nutrients to the egg cell during development. So shortly after fertilization, they also break down. Question number 12. Name a disease caused by the lack of each of the following in the human diet. Part A. Vitamin C, scurvy. B. Iodine, goiter. Question number 13. How are water dispersed seeds suited to their dispersal? Number 1. They have waterproof coats to prevent soaking by water. So if they want to be soaked, if they want to absorb water, they will become heavy and sink to the bottom of the ocean floor. So dispersal will not take place and therefore germination cannot occur. So they have waterproof tester. Next one. Some, like coconuts, have fibrous and spongy mesocarp to trap air. They have air spaces in between so that they can trap air. Now, by trapping air, it makes them light and buoyant for easy dispersal. Others have seeds whose tester trap air bubbles. So when they trap air bubbles again, they become light for dispersal. Question 14. A person was found to pass out large volumes of dilute urine frequently. Name the disease the person was suffering from. This is diabetes insipidus. Part B. The hormone that was deficient, antidiuretic hormone. Question number 15. Name 3 aspects of growth in living organisms that can be quantified to determine growth. Height, weight, length. As easy as that. Question number 16. How is the DNA molecule? Question number 16. How is the DNA molecule suited as a hereditary material? Number 1, it's relatively stable. So what happens is that it can be passed from one generation to another unchanged. Number 2, it has the ability to replicate itself during cell division. And lastly, it also has the capacity to undergo mutations. These bring about, genetic variations. And these variations, by the way, are very important because they are essential for the adaptation of organisms. Because beneficial mutations can lead to traits that enhance the survival of an organism. Question number 18. Briefly explain the role of the following in the immune system of animals. Number 1. Lymphocytes. So lymphocytes are white blood cells that produce antibodies. And of course, antibodies target and kill pathogens. Now, Roman 2 antibodies, they kill pathogens. So antibodies are proteins that specifically target and neutralize pathogens such as bacteria and viruses. Roman 3 phagocytes. Now phagocytes are also white blood cells, but their superpower lies in their ability to change their shape. So they are able to engulf pathogens, surround pathogens and then slowly digest them. Question number 19. An experiment was conducted by some students as illustrated in the diagram below. Study it to answer the questions that follow. Okay, so let's look at our setup. We have 3 retort flasks. So if we look at A, it contains dried seeds. These have been placed in a solution of caustic potash. By the way, caustic potash is potassium hydroxide. B, you have soaked seeds placed in a salt solution. And C, again, you have soaked seeds, but in this case, they have been placed in caustic potash. Part A, state the aim of the experiment. Now, when it comes to experiments in biology, and you see a solution of potassium hydroxide or sodium hydroxide included, then just know that they are probably there for the carbon 4 oxide. And the reason is because these two solutions absorb carbon 4 oxide. So in this case, what is the aim of the experiment? It's to show that carbon 4 oxide is produced during respiration. So respiration utilizes oxygen but produces carbon 4 oxide. Now, in case you're wondering, respiration, aren't we talking about germination? Yes. So in this case, if you have seeds that have all the necessary conditions, presence of oxygen, there is moisture, they have optimum temperature, then yes, they are going to germinate. But germination is a process that requires a lot of energy. So in such seeds, what will happen is that they are going to have a faster rate of respiration. Why? To provide enough energy for the process of germination. So food substances are going to be broken down in order to provide energy for the growing regions of the seeds. So in this case, respiration takes place. Now, part B, state and explain the observations made in setup C above. So in C, our seeds are soaked. So yes, they are going to undergo germination and therefore carry out respiration. But the retort flask has been placed in potassium hydroxide. Now before I explain, let us talk about the change in the volume of the gases. And specifically, I'm talking about oxygen and carbon 4 oxide. So as we stated, respiration will take place. Respiration uses oxygen. So what will happen is that the volume of oxygen in the flask goes down, okay? But the volume of carbon 4 oxide increases because this is a product of respiration. But immediately the carbon 4 oxide is produced, it's going to be absorbed by the caustic potash, by the potassium hydroxide solution. So essentially, you're going to have a drop overall in the volume of gases present in the retort flask. This causes a drop in the pressure within the flask. So you're going to have a lower pressure in the retort flask as compared to the atmospheric pressure. And this causes the solution to rise up the flask. This is our observation, and that is our explanation. Part C. What is the role of setup A and B? These serve as control experiments. Question number 20, Roman 1. Define the term polyploidy. So this is a condition where a cell will have more than 2 sets of chromosomes. Roman 2. Polyploidy is common in plants. Name the advantages associated with it. Now, when it comes to plants that have polyploidy, they tend to have increased yields. They also mature earlier. So you can harvest earlier, when you have polyploidy plants than diploid plants. And even better, they are more resistant to pest diseases, even environmental conditions like drought. Question number 21. Differentiate between the open circulatory system and the closed circulatory system. So in the open circulatory system, the hemolymph is pumped into the general body cavity. So it's going to come into direct contact with the body tissues. In the closed circulatory system, blood is pumped into closed vessels. So it doesn't have direct contact with the tissues. Number 2, in open circulatory system, the transport fluid, the hemolymph, flows under low pressure. So you're going to have blood circulating over a short distance at a lower rate. In the case of the closed circulatory system, blood flows under high pressure, so the blood circulates over a longer distance at a faster rate. Last one, in the open circulatory system, it's less efficient in transporting nutrients to the tissues. Now, in the closed circulatory system, it's more efficient at supplying oxygen and nutrients to the tissues. I want you to note this. When I talked about the open circulatory system, I did not mention transportation of oxygen because in the open circulatory system, such as that which is found in arthropods, in insects, the circulatory system is not involved in transportation of the respiratory gases. Yani, it's not involved in transportation of oxygen and carbon dioxide. Kabisa. The system that is involved in the transportation of these gases is the tracheal system. Okay? Question number 22. Differentiate between mitosis and meiosis. So in mitosis, homologous chromosomes do not associate with one another. Meiosis, homologous chromosomes associate with one another. Mitosis takes place in one nuclear division of 4 stages. Now, in meiosis, this takes place in 2 nuclear divisions with each having 4 stages. Mitosis produces 2 daughter cells with each being diploid. In meiosis, you're going to have 4 daughter cells, each being haploid. Mitosis occurs in somatic cells. So this leads to the growth of an organism. Meiosis occurs in reproductive organs, leading to the formation of gametes. Question number 23. Name the role of the following apparatus as used in biology practical activities. Part A. The pitfall trap. This is used to trap small crawling animals. Part B. A pair of forceps, picking small dangerous plants or animals. Part C. The bait trap. This is used to attract and trap small animals. Question number 24. A student was planning on how to estimate the population of fish in a school fish pond. Part A. State the method he would use. Capture method. Part B. List 2 assumptions made in the study above. So you're going to have assumptions that a person makes when they use the capture method. So they are the following. That no fish moves in or out of the areas between the 2 counts. The released fish would be able to mix freely with the remaining population. The mark would not alter the fish's behavior. And lastly, the marked fish will have enough time to mix with the rest. So these are the assumptions. Now that brings us to the end of this revision lesson. In the next one, I'll be tackling biology paper 2. See you there.